Hi friends, today we are going to see the past year NEET questions for the chapter Cell, the unit of life, and this video will cover part 2 for you. We've already covered part 1, so kindly have a go through. So let's begin with the first question here. Let's read it here. Why is a capsule advantageous to a bacterium? So this question was asked in the year 2013. Okay, so before we go into the solution, let's discuss about what is capsule. So what is a capsule? You have heard about these terms, right? Even when you take tablets, you've heard that they are in the form of capsule. So what is the work of the capsule? Is the capsule have any medicine in it? No. Say the capsule is the outer covering. Okay, if you open the capsule, sometimes by mistakenly, if you open the capsule of the tablet and see, you'll have content in it. You'll have the medicine in it. So it's the covering which gives the protection for that medicine. Certain medicines, they require an extra covering like capsules, but these capsules will not affect the body. Only the medicine seems to have the effect, but the capsules are non-injurious to the person who takes it. Likewise, this capsule, which is an advantageous to a bacterium. So what is a bacteria? It's a microorganism. Yes, it's a microorganism. We have two-way round. We have advantages from bacteria. We also have disadvantages from bacteria. So how come this capsule becomes an advantage for a bacteria now? That is a question. So given 4 options here. Look at the first option. It allows the bacterium to attach to the surface, or does it protect the bacterium from desiccation? Desiccation means from heat. Okay, from heat. Then does a capsule help in locomotion, or if it allows the bacterium to hide from the host immune system? So coming on to the point, capsule is a layer that lies outside the cell wall of a bacteria. So the bacteria will have a cell wall. So it's an extra surface that covers the bacterial cell wall. Okay, the capsule can protect the cells from engulfment from eukaryotes. So what do you mean by engulfment? Swallowing. Okay, engulfment means swallowing, right? Engulfment by the eukaryotic cells, especially macrophages, which our immune system has it. It has a large structure in many bacteria. And if you see this capsule, mostly it will be polysaccharides, a long chain of sugars. It is considered as a virulence factor because it enhances the ability of the bacteria to cause the disease. So what does this bacteria do? If you see, this capsule do, if you see, it allows the bacterium to hide from the host immune system. Okay. As I already said, this is an extracellular surface that is present over the cell wall of the bacteria. So what happens is, this prevents the engulfment of the bacteria by the macrophages, which the host system will have, host immune system will have. So it was actually escaping from the macrophages. So it means it is hiding. Okay, the right word you can put here is to hide from the host immune system. So therefore the correct answer to this question is option D. Okay, now let's move to the next question here. Now look at this question. This question was asked in the year 2012. Which one of the following does not differ in E. coli and Chlamydomonas? Now what is E. coli? What is the full form of E. coli? It's a microorganism. It belongs to bacteria, and the full name of E. coli is Escherichia coli. And Chlamydomonas, you all know, it's an algae. So what is that which will not differ in both? Okay, so before we go into the answer, we will discuss both. E. coli, as I said, it's a gram-negative bacteria. Okay, it's a gram-negative bacteria, meaning it will take the pink stain and it will appear pink under the microscope in the form of rods like this. It will appear, and it is in the rod shaped, bacilli shape. Okay, so mostly if you see, this E. coli is found in the gut of the warm-blooded organisms. They sometimes or very occasionally they cause illness. Okay, so now to tell about Chlamydomonas, as you all know, this is a unicellular green algae, right? We all agree with that. It's a unicellular green algae, and it consists about 325 species of all unicellular flagellates. And its habitat, if you see, it is found in the moist, damp areas, or even in the stagnant water. Okay, so what is the coincidence? What is something that is common in both? If you see ribosomes, no. Chromosomal organization, again no. Cell membrane or cell wall. It should be either of this. Okay, the correct answer goes to option D, cell membrane. Okay, why? Because both the organisms that are mentioned here, E. coli and Chlamydomonas, will have a cell membrane that is made up of or composed of lipids and proteins. So it's here, lipids and proteins. I'm sorry, lipids and proteins. Okay, so here if you see, you can see the gram-negative bacteria. This is how it will be under the microscope, and this is how Chlamydomonas will be under the microscope. Okay, so it will be like this only. Therefore, the correct answer to this question is option D, cell membrane. Okay, now let's move to the next question. Okay, coming to this question, nuclear membrane is absent in dash. This question was again asked in the year 2012. Okay, you're given here 4 organisms here: penicillin, Penicillium, Agaricus, Volvox and Nostoc. So any guesses what can be the answer? Penicillium, you know, it is fungi, so therefore it will have a nuclear membrane, so the Penicillium cannot be the answer. Agaricus, Volvox, they all have a nuclear membrane. Okay, so the one which does not have a nuclear membrane is Nostoc. Okay, what is this Nostoc actually? It is a prokaryote. Okay, and being a prokaryote, you all know that prokaryotes do not have they lack membrane bound organelles and a well organized nucleus. Okay, that is what. So when they don't have a proper system, obviously they will not have a nuclear envelope or a nuclear membrane over it. Right, so that is what they mean here. A nuclear what is a nuclear membrane? It's a bilayer membrane which surrounds the nucleus. Okay, therefore the correct answer to this question is option D, Nostoc. Nostoc is a prokaryote while rest are eukaryotes given here. Nuclear membrane is absent in prokaryotes, and sometimes the prokaryotes will not even have the membrane bound organelles. They don't have a proper organization of the organelles for the functioning. Okay, the correct answer is option D, Nostoc. And here if you see, to your right side you can see a picture which shows a heterocyst in the Nostoc. So this is how it will appear under microscope as spherical shapes. Okay, now let's move to the next question here. This question was asked in the year 2006. Okay, which one of the following statements regarding Celia is not correct? Okay, now what is Celia? You should know what is Celia, then only you can answer this question, right? Celia are made up of tubulin protein. What are they made up of? Tubulin protein. Okay, Celia is made up of tubulin. It's a protein actually, and Celia have a core of 2 microtubules, and around there are 9 doubled microtubules. Okay, these are not actually hair-like structures. Okay, many actually confuse cilia with flagella, and sometimes we consider them as hair-like structures, but they are not hair-like structures. And you have 2 types of cilias. What are the 2 types of cilias? One is motile and one is non-motile. Motile means it will help in movement, and non-motile means it will help in it will act as a sensory organs. So it will act as sensory organs. So the correct answer to this question is option A. Cilia are hair-like structures, hair-like cellular appendages. As I already said, cilia has microtubules. They are not hair-like structures. Okay, but they have microtubules. And if I say one can be motile or non-motile. When I say non-motile, they will not help in movement, but they will act as sensory organs. They can sense the environment. Therefore, the correct answer to this question is option A. Now, let's move to the next question here. This question was asked in the year 1995. The prokaryotic flagella possesses dash. So this question, if you notice, you're given 4 options here, and it is quite confusing. Look at the first option: unit membrane enclosed fiber, protein membrane enclosed fiber, So first two options we can understand. But what about the option C, 9+2 membrane enclosed structure? Did you ever think what is that, or do you know what is that? See, when I say a 9+2 arrangement, okay, it is actually the arrangement of the microtubules. They are talking in that manner. So 9+2, you don't have to get confused what is that arrangement. So it's the arrangement of the microtubules, okay, which sums up to form a cylindrical structure like cilia and flagella. That is, you will have 9 fused pair and 2 unfused pair. So 9 represents, okay, now 9 represents the fused pair and 2 represents the unfused pair, and these pairs are nothing but the microtubules. So that is about 9+2 arrangement. So the prokaryotic flagella possesses, the correct answer is helically arranged protein molecule. Prokaryotic flagellum is made up of flagellin protein. Okay, it is made up of flagellin protein. Okay, as we saw in the previous slide about different proteins that are present in the tubule, right? Likewise, flagellin is one kind of protein. We already saw proteins like tubulin in microtubule size. So this is responsible for the flagella protein. And this is arranged helically. It do not show any 9+2 organization or ATP's activity, anything like that. These flagella do not beat. Okay, their movement, we are talking about the movement. They do not beat, but they rotate like a propeller that brings about a backward pushing of water. Mostly the gram positive ones will have 2 rings in basal bodies, and the gram negative ones will have 4 rings in their basal body. Therefore, the correct answer to this question is option D, helically arranged protein. Okay, so as I already said, gram positive bacteria means they will take the purple stain and they will have 2 rings in their basal body, and gram negative, if you see, they will have 4 rings. Okay, so here if you see, it is the backward pushing of water. How is their movement? Backward pushing of water due to the helical arrangement of the protein molecule. Let's move to the next question here. This question was asked in the year 1993. Golgi apparatus is absent in dash. Any idea? What is a Golgi apparatus first? What is a Golgi apparatus? A Golgi complex is a complex of vesicles and folded membrane within the cytoplasm. So it is present within the cytoplasm, right? And therefore it is present in most of the eukaryotic cells which are involved in secretion and intracellular transport. When I say intracellular, we talk about the transport which happens within the system, not outside, external. It is internally. So now you got the clue what is Golgi apparatus. In which system it is absent now? It is absent, is it absent in higher plants or yeast or bacteria, blue-green algae? None. The correct answer goes to option C, bacteria and blue-green algae. As we already saw in the earlier slides, prokaryotes do not have proper membrane-bound organelles. They have no proper organization, and therefore their system is it's just a very simple system. It's not complex actually. A single cell will contribute to all the functions in that particular prokaryote or blue-green algae. They all come under prokaryotes. So Golgi complex is absent in prokaryotic cell, especially the, what do you mean by PPLO? Polypleomorphic bacteria. Okay, so here if you see, this bacteria, blue-green algae, they are all belonging to the section of prokaryotes. So it is Golgi complex will be present in all eukaryotes, okay, except in the tubes, sieve tubes in plants. You have to understand this. See, where all the Golgi complex is not, except in sieve tubes of plants, sperms of bryophytes and pteridophytes and red blood corpuscles. So Golgi complex is absent in these areas of eukaryotes. Okay, let's move to the next question here. This question was asked in the year 1992. Experiments of acetobularia by Hammerling proved the role of dash. So what do you mean by acetobularia? It's a genus which belongs to green algae. Okay, it belongs to green algae. and it belongs to the family full of Polyphysae. Okay, we have Chlorophysae, Polyphysae, brown and all that, right? It is typically found in the subtropical water region, and mostly this Acetobularia will be brown. They will be single-celled organisms, and it is also called the mermaid's wine glass. It is also called as a mermaid's wine glass, the other name of this, because the shape will look like that. Okay, the presence of hereditary information in the nucleus was proved to be the work by Hammerling. So the correct answer is option B, nucleus in hereditary. Okay, heredity. Fine. What did I say the other name for Acetobularia? It is called as the mermaid's wine glass. Okay, so what did this person do? If you see, he carried out experiments with this green algae, and he actually found that nucleus is in heredity. Okay, the presence of hereditary information in the nucleus was proved by the work of Hammerling on a single-celled green algae, which is Acetobularia. Therefore, the correct answer is option B. Now, let's move to the next question here. This question was asked in the year 2017. Okay, let's read it here. Which one of the following cell organelles is responsible for extracting energy from the carbohydrates to form ATP? What is the full form of ATP? Adenosine triphosphate. Okay, so what does this ATP do, ma? Adenosine triphosphate is a complex, and it's an organic chemical that provides energy to drive many processes in our life. We have already spoke about this. We all require energy to survive, right? Even when we skip even a single meal, okay, what happens? Our energy level comes down. That is because the production of the ATP is not happening, and already which is stored is being utilized by the body. So we will have that tiredness. Okay, we don't have that energy. Okay, what are the activities this ATP can contribute to? Anything, every work that we do, even we have to push something, we need energy. We all agree with that, right? It can be muscle contraction, it can be even a simple nerve impulse that happens within the body. All the activities in our body require energy. Therefore, this ATP is very important, and this synthesis takes place in which organ? It happens in mitochondria, and to your right side you can see the picture of it. Mitochondria is the powerhouse of our body. So this is how the image is, this is how the internal structure of the mitochondria will be. It will have a matrix, it will have intermediate space, ribosome, inner membrane, cristae. Cristae are the foldings. Okay, cristae are the foldings, outer membrane, inner membrane. So this mitochondria is also called as the energy factories of the cells, and this ATP production by mitochondria is done by the process of respiration. Okay, respiration. Okay, this respiration will produce energy. So it's a continuous activity that happens in your body. So you require only when you intake food, that energy will be absorbed. Therefore, the correct answer is option C, mitochondrion. Now, let's move to the next question here. This question was asked in the year 2016. Okay. Mitochondria and chloroplast are, and again, if you see which one of the following options are correct. So, you both A and B correct, or both A and B are wrong. So, what you have to decide? Okay. Mitochondria and chloroplast are they semi-autonomous organelles, or they are formed by the division of the pre-existing organelles? They contain DNA but lack protein synthesizing machinery. So, the correct answer goes to option C. See, mitochondria and chloroplast are of course semi-autonomous organelles. Semi means partly. They are actually not fully dependent. Okay, semi means partly, right? Now if you say semi-fluid, it is either solid or liquid. It is somewhere in between. Likewise, when we say semi-autonomous, autonomous mean they can do their activities without the help of anyone. They are not dependent on anyone. They have the authority to decide of what to do. So when you say semi-autonomous, they have the capability to they have partly the capability to do what they want to do, and partly they are dependent. So it's a 50-50 probability, right? So A is true but B is false. They are not formed by the division of the pre-existing organelles, and they do not contain any DNA, or if they lack protein synthesis, no. Therefore, the correct answer is A is true but B is false. Mitochondria and chloroplasts are semi-autonomous cell organelles, and they are formed by the division of the pre-existing organelle, and they contain DNA, but they also contain protein. So here they have mentioned they lack protein synthesizing, but they actually contain protein synthesizing machinery also. Therefore, the correct answer is option C. Now let's move to the next question here. Now look at this question here, was asked in the year 2016. Microtubules are constituents of dash. So what all the microtubules contain? Now what is microtubules first? Microtubules are polymers. They are polymers. Poly means many, so it's a long chain. Polymers of tubulin. Tubulin is a protein that form a part of the cytoskeleton and provide structural shape to the eukaryotic cell. So this is the work of the microtubules. They can grow as long as to 50 micrometer. And role of the microtubules is that in the cytoplasm, they include mechanical support, organization of the cytoplasm, transport, etc. You have many activities, but I've just mentioned a few. Okay, so what is their activity? Where and all they are present? What are the constituents of? If you see, they are a constituent in cilia, okay, cilia and flagella for the movement. Okay, then they are present in centrioles for chromosomal movement and spindle fibers for the structural organization. So microtubules are a constituents of spindle fibers, centrioles and cilia and flagella. Here they have, so the correct answer goes to option B. As I already said, see, they have three, they are constituents of three. Spindle fibers will help in structural organization. Right, so you need to know this. Without knowing this, you cannot answer the question. You need to know which activity belongs to which. Then we have the centriole. In centriole, when microtubules will act for or will contribute for chromosomal movement. Okay, and the next one in cilia. Cilia or flagella. Okay, they help in movement. Cellular movement. Okay, therefore the correct answer is option B. So to your side also, to your right side, they have explained it in depth. I've told you in current these microtubules are constituents of what and what is their function in that each constituent. Now next question. This question was asked in the year 2016. Which one of the following cell organelle is enclosed by a single membrane? We all know we have three types of membrane. One is single membrane, double membrane and A membrane, that is without a membrane, right? So first we will see the ones which have single membrane, no membrane and double membrane. Out of the four options given here, let's talk about mitochondria. Mitochondria has double membrane. Okay, chloroplast has, what does chloroplast have? Again, it's a double membrane. Lysosome, it is single membrane. Okay, what about nucleoli? Is it having a membrane or not? Nucleolus does not have a membrane. So will a nuclei have? No. Therefore, the correct answer to this question is option C, lysosome. Okay. So this is how they will vary. Now, for example, in the solution part, they have given you double membrane wound organelles are mitochondria, chloroplast, endoplasmic reticulum, Golgi body, nucleus. Nuclei doesn't have. Okay. Nucleolus is membraneless. Now, next question. This question was asked in the year 2015. DNA is not present in dash. Okay. What is DNA? What is DNA? Deoxyribonuclear sugar, right? This DNA is a hereditary material. Okay. Apart from that, this DNA, there are some organelles which do not have DNA. First, we will discuss one by one. What do you mean by ribosome? Ribosome are minute particles. We are talking about ribosome now. They are minute particles consisting of RNA and also protein found in large numbers of the cytoplasm of living cells. Okay. DNA, nucleus and chloroplast will have DNA, while ribosome will have RNA. Okay, RNA and associated protein. Therefore, the answer is very simple. The correct answer goes to option A, ribosome, because they have RNA in them. Mitochondria, nucleus and chloroplast, all the rest will have DNA. Only ribosome will not have DNA. Instead, they will have RNA, ribonucleic acid, and they will have the associated proteins with it, and they are surrounded by any membrane. They are the site of protein synthesis. To your right side, you can see the ribosome. Okay, now let's move. So the correct answer to this question is option A, ribosome. Okay, clear? Now let's move to the next question here. This question was asked in the year 2015. Nuclear envelope is a derivative of dash. We already saw. What does a nuclear envelope do? Nucleus has double membrane. We all saw it in the earlier slide. Nuclear membrane is a double membrane composed of an outer and an inner phospholipid layer. That is for the protection. Why do they have a phospholipid layer? Is for the protection. Right? Now, between these two layers, if you see, there is a lumen which connects to the endoplasmic reticulum. Okay, so if you see, it is formed during the telophase by fusion of vesicles, into which the nuclear envelope develops during the prophase. So what happens here is nuclear envelope is a derivative of rough endoplasmic reticulum. Okay, as we all know, when we say endoplasmic reticulum, it can be classified as rough and smooth. So this diagram clearly tells you it's a rough and smooth. You have nucleolus, nucleus, nuclear probe, ribosome and all that. So here in the late prophase, the nuclear envelope disappears and it will reappear in the late telophase from the endoplasmic reticulum. So they are asking us which is the derivative of nuclear envelope. Therefore, the correct answer is option C, rough endoplasmic reticulum. Okay, now let's move to the next question here. Select the correct matching in the following pairs. This again was asked in the year 2015. Okay. We all know there are different, as we all know, endoplasmic reticulum has smooth and rough based upon their appearance. That is how we classify. So I will tell you the answer. Smooth endoplasmic reticulum is responsible for the synthesis of lipids, and rough endoplasmic reticulum helps in the synthesis of proteins. So here if you see, option A is correct while the rest of the options are wrong. Rough endoplasmic reticulum, I said it is helping in the synthesis of protein. Therefore, synthesis of glycogen or synthesis of fatty acid, oxidation of phospholipids, all this will not come under this. Okay, therefore the correct answer to this question is option A, smooth endoplasmic reticulum. It helps in the synthesis of lipids. Okay, next question. Now this question again 2015. Which one of the following are not membrane bound? Okay, this is a very easy question. I think by now you have guessed what the answer is. Yes, the correct answer is ribosomes. While the rest, if you take lysosome, vacuoles, mesosomes, they are all membrane bound. Ribosome, if you see, they are not membrane bound, and one thing unique about them is that they are minute particles which are present in the body, and they consist of RNA and they are associated proteins, and they are present in the cytoplasm of the living cells. Not every system will have that. Okay, therefore ribosomes is not membrane bound. The correct answer goes to option A, ribosome. So ribosome, they have RNA instead of DNA, and they are found in large numbers in which location of the living system? Cytoplasm. They are found in the cytoplasm. They are found in the cytoplasm in large numbers. Okay. Yeah, the location where you find ribosome is cytoplasm. Now next question. Okay. So this cytoplasm is correctly in the living system, and this ribosome, as I mentioned, is very unique, and it does not have a DNA. Instead it will have RNA and associated proteins in it. Right, we all agree with that. Okay, so this is the last question for this chapter. I mean for this chapter, Cell, the Unit of Life, and today we complete part 2 in this video. Okay, so we all will meet in the next part of the same chapter, Cell, the Unit of Life. Okay, thank you.